if for those that for whatever reason may not be present, and maybe down the road if you guys have questions for your lab exam, then you can always reflect back by reaching out to me. But what I do want to say and highlight first off is regarding your notebooks. I think it's very important to highlight that some of you may not be aware about the importance of having detailed figure captions for your results. Remember, we had four types of results from last week, technically pre-reading week, and that included your streak plating, right, for the figure that you would have drawn down for your observations of your streak plate from week 4. That includes as well conjugation. Actually, I'm going to move this up because you may not be able to see it. That includes streak plating, conjugation, transformation, and hopefully you also grasped it for your plaque assay, which is due for your FLR, due on March 8th, 10 p.m. So I just want to highlight a bit about that. We'll talk about it now because it's important for you to realize before the GT assignment is due. First off, and just by the way, folks, I am keeping an eye for those that are paying attention, those that are, be it on their phones or are not paying attention. You don't want to lose out for your lab performance, and that is incorporated grade for this class. And just bear in mind that when it comes to, as I said out in the inbox via Quirkus, everyone hopefully saw that message. Has anyone not seen that message I sent out on Sunday? Any questions regarding it? Because it's very important that you realize one is the quality of your notebooks for the lab notebook grade. Another is your lab performance, which includes your proficiency during the lab, your participation with questions, and overall your interactions and ability to complete the lab proficiently with additional grading in mind. I do also want to highlight the importance of respect to your group members, but also to me. I do notice some individuals or showing a level of hostility, and I actually have the right to excuse you from the lab, meaning that you get a 0. So I just want you to understand that because it's very important that students and TAs are respected, and that's actually part of the code of student conduct here at UFT. So just bear that in mind, please. And so I just want to highlight that because I, for one, aim to be very respectful and considerate for everyone here. I want all of you to excel and succeed in this lab throughout every practical that we have, but you just have to bear in mind that you're not only interacting with yourselves positively, but also to myself, to the content that you're dealing with today, and also bearing in mind the implications of your results, as well as the implications of your behavior with others inside and outside of the lab. Any questions before I move further with today's presentation? And by the way, I'm not a harsh person, but I have to highlight this because I've been noticing this a few times and it's unacceptable. It's unacceptable to be disrespectful to your peers. And I've noticed this as well for bullying, and it's unacceptable. So I just want that to be highlighted. Please bear that in mind, folks. Because I can share another warning if it's needed. But if I see it intolerable, then I'll have to speak with you directly, with perhaps Dr. Brent. So just bear that in mind. Moving forward, and I do want to say in a more kinder note that congrats, folks. You're halfway into into the week, in general, into the practicals here at BIOC17. So congrats. We're into week 6, and we'll conclude at week 10. So just bear that in mind. We have a few presentations. We have 3 more assignments. I talked about the plaque assay for your FLR. Remember, for details, I've also shown them on the inbox or on Quirkus. You'll also see it on Dr. Brunt's outline for the formal lab report. But briefly, you have an intro, results, and discussion, including references section. Bear in mind that you are as detailed or as cognizant as to the details described in the outline, as well as the tips I provided. Try to be generic and like going down from generic details about plaques and bacteriophages all the way down to your research question and intro. And then for you don't have materials and methods, but for your results, you want to encapsulate, especially for your figures, the details that have been described and the conditions that you use to treat, i.e. the specimens of interest, or get your results from your methods. So that's something I want to highlight because I've seen it from some of your lab notebooks. Some of you have great detail, which I greatly appreciate, like labeling specific panels and having a detailed figure caption with highlights of each segment. But it's very important that consistently each member of this practical does the same, because that's something that's going to be considered for your FLR grade, or your FLR in general, for its quality and assurance. And then describing the results there as well, after, in between figures and before them as well, and the results that you would have encapsulated, i.e. like the PFUs for your plates, and the calculations for them. They're also, if I'm not mistaken, highlighted somewhere here, but you can also look, and if you have questions, feel free to reach out anytime. And discussions also are included, just briefly highlighting your objective, the limitations as well that you would have obtained, maybe considering the different limitations that exist from your methodology, and the extensions and implications of your results through and beyond your discussion. And just make sure that you have two primary articles in your references, but you can have more from your intro to your discussion. Any questions regarding that? Okay, so let's go straight to Lab 6. I just want to put as well a preface for timing for you guys. We are okay with timing. It's just something that I want everyone to bear in mind, that within your groups, try to essentially have someone with a timer to keep track of time for the rest of today's practical. I'm just going to highlight a few details here for timing. For exercise 1 and 2, each should be done in the next half hour. So i.e. it should be done both exercises in 1 hour time. So maybe hard to see, but 30 minutes. Exercise 2, 3 minutes. Exercise 3, 20 minutes. In case you also need to catch up, it technically can be, so it will be more like to have a buffer just in case you need time, but it can be about 15 minutes. If anyone can see it from the back, from either side, or even from the front, let me know. It can be difficult. 4 and 5, both are done in 10 minutes, and they're pretty much done with a few details that are provided on the slides. And with that, hopefully we can be done with a good amount of time, but I just wanted to keep track of timing for groups, so that way you don't have to feel rushed through that. Any questions regarding that, Andy? Any questions? Any questions? No? Okay. So first off, we know from the lab manual, actually, let me see if timing. Okay, so I'll skip ahead and use the slide for now. First off, let me show you with what you need for, once again, today's lab. You have different materials from that side. There should be 2 groups that need pipettes that they can grab down there. There is pipette tips as well with the glass pipettes that you'll be using for the microplate reader. Alternatively, you could use the plastic ones that you have at your disposal, but we have many other test tubes present as well. And so I want to see if I could turn on that projector so you guys can see as well. Okay. Okay. So now it's 9:45. So I'll just walk you through a few things. For today's exercise 1 really focuses on the microplate reader. We did this before, but in a nutshell, based on what you see on slide 3 and based on what you see in your lab manual, we'll be using different wells per group to analyze the different conditions. So germicides essentially are different substances that essentially can kill bacteria or germs, but they have different levels of severity. And so disinfectants, you probably are aware of, that are separate from antiseptics, right? Consider the location and where you actually add them on. But I want to just highlight a few other things that we have the liquid product that we need to create here. We have the disinfect this is for the catalase test on clean water. We I think here, also here. I think tubes are available. Slides are available for 3 to 4. I think slides 3 to 4. And we have well same line. But I just want to go over this briefly. That in your groups, so recall a few things, right? 1, 2, 3, 4, 5, 6. Can everyone see it from this side, by the way? Everyone? Okay. Briefly, essentially, you're going to be one of each of the. You're going to be one of the following groups with each disinfectant or antiseptic. So for group 1, you'll be dealing with bleach. Groups 1 and 2. And then groups 3 to 4 will be dealing with hydrogen peroxide. Groups 5 to 6 will be dealing with ethanol. And we have them available. But what I just want to highlight is that for each odd group, so group 1, you'll be dealing with E. coli, group 3, E. coli, group 4, E. coli, group 5, E. coli, and then group 2, 4, and 6 will be dealing with S. epidermidis, Staphylococcus epidermidis. You guys know why we're using these specific bacteria? Did anyone show a raise of hands or have any thoughts? Think about your unknown assignment. One was gram positive and one was gram negative. Which one was negative? E. coli? Which one was positive? S. epidermidis, right? So think about that because it's actually important considering the morphology and the differences in their cell wall or cellular composition. Because for gram-positive bacterium, they have a thick peptoglycan layer or wall that will be additional. Think of it as a secondary, a thick secondary layer available for them. So think about the roles of the disinfectants versus the antiseptics that are added onto the surface. So that way you'd be able to understand the appropriate and hopefully most efficient or effective treatment for these different bacterium. And then essentially at the different propensities or concentrations, which I believe you guys have as well, we'll be adding them into the microplate. So I just want to highlight these are your saline tubes. So looking for something. Yes, perfect. You guys have it. Sorry, it's a blue one. I was just having a look. The blue ones, yes. So they're already aligned. They're already there present for you guys, which is great. I just want you to bear in mind, though, what you need to do. Make sure that you take what the saline tubes that you have available right here. You add in. Let me grab one of your liquid drops. So look at your red tray from your tubes. For example, follow a aseptic technique once again. This is E. coli, for example. Add in as much, so you can actually pour it, but it's important not to fill it up beyond the tip of the agar. So the agar side, i.e., just go up to maybe an inch beneath, so that way it's readily diluted. Not too much, but readily concentrated so that you don't over dilute. Because that will be important for taking absorbance readings when we deal with and then accordingly you'll be able to do the other the appropriate exercises in due time. Any questions regarding it will be essentially, so when you do, yes, so it won't be, you don't have to put the entirety, like I would not recommend anyone piercing through the agar because you have once again, like the common swabs that you need, you have tweezers for exercise too, but generally speaking, you should use the material that you have available with you, and that should suffice. But yes, thickness should be like milky white essentially, but don't pierce through the agar. So that way it doesn't add any unaccounted for defects or artifacts. Any further questions? Any questions? Okay, so let's go straight ahead. Both, I want to, let me just go. Oh, yes. And incubations will be at room temperature. And in terms of updates, just I would say focus with what I've talked about thus far. Use based on what we've also talked about, the positive, negative controls, which should be highlighted. They include essentially your bacterium, right? Your bacterium of interest, and pretty much saline water. So that would be 1 mil. And so just to highlight this in your groups, remember group 1, 2, 3, based on your numbers, the different labels that are shown here. And it's important that you replicate this because your groups essentially go down row by row. So just bear that in mind. Group 1 goes down all the way here. As an example, you have your blank, you have your positive, then your negative here, then your low to middle or medium to high concentration of your disinfectant or antiseptic. So just bear that in mind. All are 1 mil, but you just need to make sure that you're controlling this well because otherwise you run the risk of not performing the mission for the microplate effectively. So when you're essentially, I'll take out the microplate meter now, and hopefully in due time, you guys can turn on your Bunsen burners. Remember aseptic technique with the bacteria that you have available in you. And just give me 1 moment before you officially begin. No, sorry, I mixed them because I'm thinking about 2 other exercises, or my mind was I was just a bit occupied, but something to note out for your liquid draw, that's where you're focusing on for exercise 1, because after all, that's by the way needs to be resuspended well because you have it already dense. But for exercise 2, when we're dealing with the plates, think of it just like how we've done it in the past. With spread, not spread plating, but essentially with your solid agar. So if you look at your red trays, solid agar, exercise 2, liquid agar, sorry, liquid cultures with the exercise 1. So look at these two students, you'd like to have them with you available. Just make sure to resuspend regularly or abundantly, so that way you can ensure a turbid reading is available for the microplate reader. I see a question.
Are you going to do the original, like the first baseline reading, or are we doing that on the
Microplate, it'll be taken care of. Don't worry. So I know some of you may be wondering with regards to the generation time and for this exercise for today, it's important that the baseline is going to be 0.06. But that being said, the ratios for equalization is something that you guys don't have to worry about, i.e., the results that you see from the microplating meter should suffice. If anything is required, I will update you on Quercus after the lab for calibration purposes. But you guys don't have to worry about that. That should be taken care of. Any other questions? Questions? No questions? Any questions? Yes. If you have them, what is that? Use them. It is technically okay. Yes, you guys can begin. I want to give you about up to 10:30 to complete this exercise. And that should suffice for timing. If you guys want as well, You could separate tasks to focus on exercise 2 as well to make use of timing. And then likely we'd be done 10 by 10:45, both exercises. And then I would say the last 4 or even 3 are more easier to do. We can even do them in synchrony as an entire group. But I would say focus on a microplate reader. And then we'll talk about plaque assay afterwards as well.
So are we doing this step or we're setting our methods
I'll be taking care of that, so don't worry.
So we're just starting.
No.
Yeah.
Of that into.
Before, do we just bring the two over?
Once you're done.
By the end.
Yeah.
Oh, okay.
Like when I mean done, I mean like you're pre-suspended, and like you've added in the appropriate disinfectants or
I'm like the
for exercise.
yeah. Okay.
So like follow up with exercise 2. I think I'll just try to copy this so you guys can see it as well.
So just do this at the end. Like, don't worry about it.
No. I would recommend doing this now.
So then, do I just bring the two over and just
But technically, you want to essentially, you need to incubate them. So that would be done in about 15 minutes.
like the liquid?
To put it before.
Yeah.
But remember, you're incubating them with your
I think this was the baseline. It's not baseline.
not for the baseline. Baseline, I'll be doing.
Oh, this is clear with it.
Are you saying, just to confirm, are you saying it with the antiseptic or disinfectant or without it?
Oh,
Without it, yeah, I'll do that.
okay, because I
It's
thought
okay.
what's it called? The manual was like first do this and then add it.
I'll be doing
Oh,
it.
you're doing okay.
That's okay.
Okay, okay.
No problem. No problem. Anyone having problems? I'm hearing a lot of beeping, but I don't know where it's. Oh, century? Okay, perfect. So, you actually have it on your, it's a separate one. So, try not to confuse the H2O2 hydrogen peroxide here. It's for the catalyst type, for the middle bench. Each group that has a particular disinfectant or antiseptic has thicker blue cylinders or conical tubes already on the bench. You also have canisters that are labeled wide and out. This one is an example here. One more. Before you open them, you use the regular liquid.
Did you just? Yeah, if we have things that's the culture of the whole. And so each one talks about that, the one that like the whole.
Before spinning down, I just want to highlight, actually first off, if you guys want to find out the absorbances, 0.154, 0.576. I know I said 0.06, small error. This is essentially the baseline of both absorbance readings that you would get for ideally your bacterium. So just take a note of that, because for your microplate reader, we can essentially artificially calibrate by the ratio that would have been adjusted, or you could do 1:1, but I would prefer an accurate reading when we're done with absorbances is to essentially divide your results by the difference. So technically, make sure that the centrifuges are set to 5,000 RPM. 2,000 is actually too low, but typically the other line was saying that the they found that it was pretty brought down, especially form of pellet. If you guys want, I can adjust it now.
Thank you. Okay.
How long do you guys want? That results are labeled. That we have the different ones. So sometimes you guys are good. So I apologize. Sometimes within the lab, there are times where, as we're one of the first practicals of the week, that the lab techs may actually give up to date or new advice. So that way you guys don't have to repeat certain processes again. But I'll try to do what I can to make sure that you guys are not limited in timing during that moment or throughout the practical. Just I want you all bear in mind that you have 25 minutes to complete exercise 1. I highly recommend, as you wait, to have other members of your group focus on exercise 2. So that way, hopefully, both exercises can be done by 10:45. Following this, the other portions of today's practical will be much easier. Trust me, it'll bring back hopefully good memories when you did some staining in weeks 1 to 2. You should have watched that. And just to clarify, I'll be doing this as well for you guys, but I just wanted to. Oh, it's not working. Okay. So continue as you're going, continue as you are, and I'll update you if needed. Yes. It's something. Folks that are exercise tutorial, you'll find here once again. We have forceps, tweezers, we have the antibiotics. Phase one, with for this, essentially, which takes out, then inwards, and then you need to bring back the entire a vial back to where it's located. Each of them are here for all five, streptomycin, tetracycline, ampicillin, and then this is for the other half. Each bench, that is very much the pilot. On one end, this actually push from one end, the other side should open up. Then you should be and then when you come to your floor, you need to place we need to when you're following the floor adding on to your you're going to be adding remember you added the rest of but essentially know where they are and then they for that's something that's kind of pretty much well let me make sure for the this is another thing. Folks, for antiseptics, antiseptics, disinfectants, they're all actually on the right-hand side. So they've actually been placed there. Groups that essentially have to replicate this will probably be in the reverse order. Just grab from one of the groups the necessary antibiotics, antiseptics or disinfectant concentrations that you have or need, and they're going to be added in all together for the microplate and on the table in the same slide. So if you have questions, feel free to look. Remember, folks, after 5 minutes for centrifugation, your pellets will be a bit small. So just bear in mind that once you're supernated, you're not sucking up too much. Go with a smaller volume if needed from your tube, and you want to ideally just leave a residual amount before continuing further. How's everyone doing so far? Good, good. I got a few nods from group 3, group 1, group 2. Are you guys okay? Yeah? Okay. 4, 5, 6, how are you guys? How long do you guys think you'd be complete for preparing for the microplate reader? $10 minutes? 15 minutes? Recommend you guys trying to budget time wisely. Think if the next 10-15 minutes you should be able to complete the microplate reader and how to prepare for adding in into the microplate reader. And then the other 1 to 2 members of your group should focus on the antimicrobial susceptibility test plate. We'll talk about that in due time, but I just want you guys to all concentrate. And around 10:30, I'll talk a bit more about exercise. If you have because you could take a fine actually at the end. Thanks. So just bear in mind, folks, you have about 5 to 10-ish minutes. Just a kind reminder for your pipette glass pipette canisters, make sure you flame them with your pipettes in advance and afterwards to close them accordingly to maintain aseptic technique. Yeah. Yes. Just I would say take one vial at a time, only one piece. So take you should take one antibiotic of each of them. So each of the small white tubes, take one of them, and then you should be able to do it. Just bring it to the front. The patient
This
like another question? You're incubating your germicide essentially treated colonies or cultures at room temperature for 15 minutes. So make sure you have a timer for it. You guys should be there in due time. And then in the meantime, we could go in a few minutes into the antimicrobial susceptibility test. But follow through with the lab manual to see what your group can focus on thereafter. Can you place it on the rack?
Yeah.
Okay.
We're not doing 37.
No.
I think this is one.
Yes.
Oh,
Click it.
with the pipette.
You can even click it on the surface of your.
Oh, like the.
Sorry?
The tube is.
Yeah.
Okay.
No problem. So when it comes to resuspension of your epitubes, make sure, folks, make sure that you resuspend your bacterium at every time, even including with the germicides. Very important to ensure that you make a readable concentration and influence and effect of the germicide of interest on your bacterium. So just bear that in mind, folks. It should be an option that give me 2 minutes.
It's too heavy. It's so wonder.
Yes. Technically 3. This is the last one. So technically there's 3 thirds. One thing. Think, give me a moment.
But the third turn is the entire process.
No. Technically doing all of them. So all of them are going to be. So when you put still do the entire.
So the entire plate.
Or you're going to do it like this.
The first one comes in. Okay.
You can ask them one. Oh, yeah, it's 3 in here. So maybe, the antimicrobial susceptibility test. Please make sure you return back the vials with the discs back to their original location.
You guys have one that's like a meter.
Please,
And then just like, and then go all the way around. And then just.
Folks, how many of you have not done incubations yet? Please raise your hand if you haven't done incubations yet with your germicide. Can I get a count? Group 3, how many more minutes are you waiting? 7 minutes? Okay. Are you guys working on the microplate? Sorry, the antimicrobial susceptibility test? Good job, you guys. They're all labeled, by the way? You're good. Or just single plate? Single plate, sorry. Yeah. Is it labeled? For each of them, yeah. What the sections? No? Group 1 and 2, how are you guys doing for incubations? And exercise 1? Okay, and exercise 1, you guys are doing? Okay. How many more minutes until they are? Or actually, I'll ask them. Group 2, how are you done with exercise 2 or 1? Is the member of your group doing exercise 2? Perfect. Okay. Okay. All plates are labeled, right? Just to confirm. Okay. If your group is done, just give me another 2-ish minutes, and then hopefully, we'll go through the antimicrobial susceptibility test. If you guys are also done, you could grab your plate and I'll talk about that afterwards. One moment. If you guys can actually align them together, all in one row, that'd be.
Where's the values page?
Yes, I updated them. The other ones were from the previous lab, with the differences. But generally speaking, I'll talk about that now, actually. So folks, as you're waiting now for incubations, everyone paying attention? So I just want to make sure everyone's on the same page. So then we are likely done at least one exercise. Likely exercise 2. All groups done exercise 2, please raise your hand. Done? Group 5 as well. Are you guys almost done? 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 Okay. Groups 2 and 5, how long do you need for exercise 2? Okay, okay. So I'll give you guys about maybe 3-ish minutes. So now I just want to give you guys some insight as to what you would do for calibration. So has everyone, by the way, seen my the files uploaded for the generation time microplate absorbance values, that sheet? For practical 2, we have them already uploaded. They were actually Stephanie highlighted to me that they weren't technically visible to you guys. And I believe someone else, maybe Krista or someone, Funmi, I believe one of you also highlighted this to me. Technically on Quirkus, they all were given published notification, but the entire file wasn't my folder, which is why you may have not seen it before. What I just want to highlight for you guys is that when you see these results, start preparing for them because you have the generation time assignment coming up in 2 weeks' time. As well, for the results that you have for like the plaque assay, we'll talk about that once again, just to highlight for your FLR, due on March 8th. But when it comes to now the calibration of the results, once your incubation's done, when you're ready to now add them into the microplate reader, I want you to keep these values in mind because for calibration, what you're doing is this. You technically have 2 different types of bacterium, right? So the idea is that if, for example, the absorbance readings are generally higher, let's say 2 times higher for Staphylococcus epidermidis versus E. coli, then you want to control for that variability. And what we do is essentially take the blank or baseline after blanking of the initial absorbance values, which I have here for our lab. The previous lab, I used their values, but I wanted to make sure they're replicable for you guys. So that way you can stay assured with the values that you receive. But I just want you to understand you have 2 options. I would highly recommend essentially taking the ratio, meaning that let's say you have absorbance values that are like, let's say 1.2 for Staphylococcus epidermidis, 1.6 and 1.8. And this is in absorbance values for turbidity, and that's okay. As you probably saw for the generation time assignment. But I want you to understand that if you wanted to then control and to correct for the values that you see, let's take the Staphylococcus epidermidis, what you do is essentially multiply by E. coli 0.618 divided by 0.688, and that will essentially give you your reading for the corrected Staphylococcus epidermidis absorbance reading. And that would be at each time point, actually. Technically, I like this result because it's more reliable. You actually are controlling for the absorbances. And so I would say if you can, you don't have to do it now, but during your post lab, at least take note of it so that way you can correct for the values accordingly. Any questions regarding this, at least? Yep. 1.20. So I put this as a streak, so it's all one line, you're not dividing it. Technically, it's 1.2 times 0.618 divided by 0.688. So it's essentially, you are accounting for, if let's say this is the absorbance, okay. I think it's going to go away. So the idea is that you want to multiply or correct your values based on the previous or the lower absorbance value. So here and at baseline. So keep in mind that 0.618 is lower than 0.688. So to correct essentially for the overestimated values that you see for Staphylococcus epidermidis, you're going to correct by essentially taking the fraction of essentially the relationship or propensity of 0.618 to 0.688. Essentially, it serves as a control because if you did the same for the baseline here, you'd actually get 0.618. So essentially, the idea is that you're correcting from the baseline all future values for Staphylococcus epidermidis by this fraction here, which is 0.618 divided by 0.688. So just essentially multiply all values. You could do this on Excel and just pouring through, and it will make things easier. You don't have to, at each time point at least, it's going to take over like 5 minutes to do. But if you have questions, let me know. Yes. Yes. One minute. Actually, I realized she said it first, actually. So yeah, what was your question? No. For E. coli, you leave it as is. I left it blank purposely. You leave the values as is of what you see on the microplate reader. We're only correcting for Staphylococcus epidermidis because it's the larger value that may be overestimated from the baseline. So that's why we're correcting it by a fraction here, 0.618 divided by 0.682. Just has like raw data values for Staphylococcus epidermidis.
Like those are
Just examples, yeah. I just wanted to make it a working exercise because it'll be less than 1.2 once corrected. And then the same exists for the other values that we see. They'll be lower than these raw values uncorrected for calibration. Sorry. Yes, that's correct.
The value we get, we don't have to do it.
No.
But with the but we're gonna get to absorbance value and then.
Yes.
Okay, so.
Okay. I'll just. Is this for? No, this is for this exercise. Exercise one. So essentially, a baseline. Technically, you can see that there's a little bit of variability. So the idea is you have two options. Technically, you can consider them nearly analogous, starting with the readings, or which is really scientific. Really easier for you to treat it as. But I would prefer that you actually compare the results according to baseline, for example. If it starts off lower, then if you see a substantial drop, let's say for Staphylococcus epidermidis, you know that drop is going to be more greater than at E. coli. So let's say if we look at 0.2, right? 0.2 is farther Staphylococcus epidermidis than E. coli, right? So the idea is that depending on the germicide that you're using, you would assume that it's more sensitive for Staphylococcus epidermidis than E. coli. Because even though it test cases, so you're just basically putting one. Yeah, exactly. So if this is too difficult, if some of you find it difficult to essentially do the calibration, it is okay to make note of the baselines at their original values, but it is something that that you want to nonetheless compare in your discussion. Like let's say I brought this up to Jianpan as a great example. Let's say your germicide concentration leads to absorbance value to 0.2, then that drop for Staphylococcus epidermidis is more greater than for E. coli. So you want to describe then the sensitivity you would expect for your germicide, let's say for gram-positive bacterium versus gram-negative bacterium. If anyone has questions on that, feel free to also ask me at the end of class. Yes.
Like the advice for making it. So just making it.
So, folks, I'm going to borrow. So, folks, I just need to make sure you guys are almost done for the microplate reader so we can begin incubation. Which group has done the 15-minute incubation for the in prep for the microplate reader? Yes, you guys are done. Any other steps before you're ending it? Yes. But apart from that, anything else? Like, are you guys missing anything? If you guys have questions as well for exercise 1, I know I gave you the opportunity to go into exercise 2, but if you guys have further questions, let me know. I'm more than happy to answer them. But don't focus too much on the incubation at 37 degrees or on the setup for the blank. I've already done the blank for you guys. I just want you guys to get ready for the positive and negative controls. So that way we could get ready. Just before I do, I think I'm gonna make this easier because I'll do some questions. Ask away. What was your question?
Like for the lab report, we have to compare between two groups. Right?
Then I'll talk about remind me if I don't.
Yeah.
I would say 1 hour.
Because the plaque assay that was posted in Focus is outdated. So
That's okay. That's okay. You can use it. You can make use of the podcast results. The reason I appreciate you guys kept yours because there's some groups that maybe before reading week weren't able to take photos or record their values. So I wanted them to at least start prepping for the seminars.
okay.
No problem. You guys are doing great. Keep it up. If you have questions?
Sorry. For these numbers we're discussing, were you talking about today? Right?
Any other questions?
No, just finding out. Like, basically, people who are going through this, like, guys, like,
Please pay attention, folks. Yes.
My careers. And then we didn't leave 400. That's not the end. That doesn't get rid of the. Then
That's okay. That's okay. 15 minutes. Yeah.
Can I?
This moment, I will just type that up to 0.5. That's pretty much 50 microliters. Take out pretty much 50 microliters and keep the remaining because that is the actually if our concentrations in the past just think about that. But on the same at the same time, yeah, actually that should ask. Any other questions? That's it. Folks, if someone, if a group has everyone attention for exercise 3, folks, I need attention. I'm just looking at the slide as well. Folks, very important. Please remember to bring back, because this is often something that we may forget. Please bring back all materials that you're not using to the middle bench or their appropriate locations. Perfect. Just bear that in mind, folks.
Wait, where are we? So after the 5 minutes, it's gonna the walk. After the walk, you're putting something down again. And then for adding all of them. Yeah, so then you're adding nutrient loss to them. And then yes. Let's give it. And then report the value. So.
I wanted to make this easier for you guys to fill other roles, but you guys are already hopefully near the end. I had like when I got leaky, so like no matter how something is how that could go like you're complaining. And you're asking.
Technically, yep. They're very similar. So, in the plastic ones, yeah. But I just want to make sure you guys are ready for that's those findings is on here if you've already done. If you haven't, I guess considering that we're already talking about it, just in case like cross contamination,
Then we're gonna. No, I do trust it. I do trust it. We had to use for the other, not for this. Well, actually, how are we? Spreading on and also the special for the two. Ready?
Let me share this. Everyone, how's it going so far? I'm just looking at.
Okay, folks. So I just want to get an update how each group is doing. I'm going to ask you guys all per group. Group 1, which exercise are you done and which one are you on right now? Okay. 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 So we'll talk about it soon. Group 2, are you done exercise 1 and 2 or are you working? Okay. Okay. So just as a kind reminder for folks, if you're done for exercise 1, please prepare yourself to actually place your tubes with their respective volumes in the correct locations based on the slide behind me into the microplate reader as soon as possible. Ideally, in the next 7 minutes, I should have all groups include all of their wells or fill up the wells accordingly into the microplate reader so they could be added into the microplate accordingly. Sorry, into the reader online or to generate it online. Now, can I actually have groups start doing this now as I continue speaking? We'll turn on the function burner. You guys maybe bring your tips. So folks, you can also bring your pipette tips to make it easier. Maybe one bench could actually keep their tips here for some time. And then they temporarily return back, if that's okay with you. 1 minute. Sorry, I don't want to get you. So can you get your 3 group 5, but just so you're aware, remember your steps based on the given conditions. The blank is already filled for you guys. So just bear in mind that for group 3, this here, you're going to be doing a positive, a B, C is negative, D, E, F, try to do and I'll get So in the meantime, as groups go in towards placing in their appropriate, and please folks, bear in mind that you're paying attention. Rahma, can you close the door? If it's okay. So I just want to make sure you guys are paying attention because especially for the microplate reader, you guys are going to be doing most of the work. So it's very important that you're paying attention to your rows and the letters or labels of your tubes. Remember, the labels go from B to E. I've taken care of A, but I need you to bear in mind that you're following the correct order because those are the values that we're using post-lab for your results. Now, if we move to, and any other questions regarding exercise 1, can I expect the tubes to be added in the next 5 minutes for everyone into the microplate reader? Great job, you guys. No problem. If you guys need extra tips or a new beaker, just let me know. I can more than happily get one. So I'm going to continue now. Groups 4 to 6, please make sure that you're able to in the next 5 minutes, as well as 1 to 2. Groups 1 to 2, please make sure that you add in your appropriate tubes into the wells of the microplate reader. If you need help, let me know. More than happy to support. But you just need to make this very soon.
Yeah, we did. No, from him. What is that?
Let me see. That's fine.
Just about which bacteria we're supposed to look for. Because in the manual it said that we should do solid and spread it on the disc, and then also solid here to spread it on the slab.
Let me.
Okay.
So, folks, I'm just gonna answer more specific this week material for now, and then for upcoming or previous assignments or assessments, we could talk about them afterwards. But I just want to highlight, first off, I see 2 wells or 2 columns complete. We're waiting for another 4 groups to add in their wells or tubes into the wells of the microplate, so that way they can be read accordingly. So please start to do this as soon as possible. In the meantime, some of you also asked me about the viable counts from last week. If your group hasn't taken them already, I'll take them out for you, and you can take them out in your groups as you wait for timing. Just remember, aseptic technique. I actually want to be here when each group actually adds in their tubes. So in the meantime, if some of you are waiting or have questions, and actually this was a common question asked, exercise 3 and 4, you're looking at different traits. One with regards to penicillin ring, and if it would be able to be essentially lysed via beta-lactamase, an enzyme that is often used in antibiotics. So the reason of you using the solid agar or broth culture is that you would still use the saline liquid or saline, essentially the salt water, into your solid agar tubes, just like how we did for exercise 1, right? Or sorry, for exercise 2 with the antimicrobial susceptibility test. So just repeat the same process with your 2 tubes of E. coli and Staphylococcus epidermidis for exercise 3 and 4, adding in just enough liquid from the saline tube such that it reaches just below the tip of the edge of the slant. So if this is E. coli or if this is S. epidermidis, you repeat the same process. Make sure that your swab is dense with bacteria because otherwise it may give you inconclusive, essentially your drop or the amount that you add in. Swab works as well for this CF cephalinase test. But essentially the idea is make sure that you take a readable amount, that the broth is essentially concentrated with your bacterium within the liquid portion that you've added in the saline water, essentially. No, so I told you about using the liquid, right? I said solid, right? Yeah, so for exercise 3 and 4, the idea is that you don't, because it's solid, right? You're adding it onto either, like, as you see behind me, the disc. The disc, essentially, you want it to react readily if it's positive. So ideally, you want to actually add it in your solid agar, liquid, saline liquid, like the tubes that you see there, to essentially make it liquefied, make the bacteria liquefied here, and that way you add in essentially like a very small amount into the disc of food. The same applies for the catalase test. It's like smearing from the past. So just follow the same steps, and you could use a swab as well. That's also okay, but just repeat the process. Solid agar works, and it shouldn't be too much of a difficult task. Actually, mine's thinking 3, 4, and okay, continue. That's gonna be just done on slide on the slideshow. You guys don't have to. I will show you. Don't worry about exercise 5. On the
Yeah, I've been emailing everyone.
Since I don't run the risk of bacteria coming in, so negative, positive? Yeah, it's positive and one negative. You guys already did it or no? No, we didn't. Yeah, positive and negative.
And then increasing in concentration.
Yes. So essentially, you're four, right? Yeah. So do you guys know how to do it? Ideally, don't forget recess. Yeah. But essentially, I'm just placing on closer to you. Closer to the person. Essentially, they can be particularly open the house. The idea is that just repeat this process. It's like one, like okay,
Yes.
Often then you find that a pellet may exist. Then before you actually go into your well, you press it in here. If you this is. I'm wondering if people made a mistake because the color comes out Yeah.
I mean, I guess we can just go by that and then the other groups can check it.
Yeah, but the thing is, in the land, see the other way around? Is negative before positive? Yeah, like, okay, let me just confirm, so they don't, there could be. But you would just do the same process. Keep it as here, you can load it, I'm just gonna ask you. Use 5. Group 5.
You're holding it.
So continue as you are, yes.
Comments fans to you.
So now that many of you are likely near done, most of today's lab, I actually want us to just briefly touch on the hand washing exercise. Does anyone have questions regarding exercise 3 and 4? Does anyone need me to clarify anything? You can use either the liquid or solid agar broth. If you're using the solid agar, make sure that you've added in saline water such that you're able to liquefy the bacteria before placing them into the disc for cephalinies, or doing the bacterial smear for hydrogen peroxide. By the way, because these results are readily available, please make sure that you're taking note of them in your observations for this lab. So folks, remember that you're making a record of the results for exercise 3 and 4 today. Make sure, folks, once again, that you're taking a recording of exercise 4. Let's say for the beta-lactamase test, you see a pink color. So think about like what we've talked about in the past. Describe your results, annotate your results with figures, and describe your discussions or conclusions. This can be done even now within your post-lab. So just start preparing for this in the meantime. The same applies for the beta-lactamase, for the hydrogen peroxide. If you see bubbles, then that's an indication quite readily, by the way, that it's catalase positive. So you need to make note, just like how we've done for the staining exercises of weeks 1 to 2, that if you have gram-positive or gram-negative bacterium, then you also want to annotate within your conclusions or discussion that Staphylococcus epidermidis, let's say, is catalase, either positive or negative. So describe these values accordingly, and that should help you prepare well. Catalase also is an enzyme like beta-lactamase, but they have different functions as you've seen from your lab manual. I'm going to help you guys. Group 2 is the second column. Just make sure that when you so you go to this one. Now, positive. So switch it to have a positive, right? Perfect. So now you know how you can orient. Oh, sorry, into the second. Just make sure that 1 minute on each. Take your time, but when you go, or like try to do it carefully because you don't want to take out like you don't want to press liquid out on the surface, but do it in the well itself. It'll be easy. Okay. Just make sure before you do so to resuspend in case. Perfect. Okay.
Thank you.
No problem. If you guys need help, let me know. I'm just gonna briefly go over hand washing, as this is actually I forgot to mention this. Some of you already took out, and I'm happy you guys did this. For the plates, for your viable counts, for generation time, how many of you, by the way, are still waiting for exercise 1 to be complete? Raise your hands. One group. Any other groups waiting to complete exercise 1? Okay. Groups. Okay. So in the meantime, hopefully the same 2 groups are done. The other 2 or 3 tests, exercises for antimicrobial susceptibility tests. If your group is done, by the way, and you forgot to add them into the incubator, you place them here. All 6 are placed, labeled, which is great. But now when it comes to the generation time assignment, you need to be able to count the number of colonies in plates between 30 to 300 colonies, essentially. Then take the dilution factor, report them based on the time points and the average of each time point. Then let me know your results because we're going to record them and I'm going to help you upload them onto Perfect. Any questions regarding that at least? Which groups have not taken out their viable counts for the generation time assignment? 1. Krista? You did not take it out. You did? Are you guys counting them in the meantime? Or already done that? Groups 4, 5, and 6, have you taken out your viable counts? Yes or no? Group 5? Group 5. Yeah. Have you guys taken out the plates?
Yeah.
正的负，然后第二那个印度我把你了，对啊，我们两个一起 positive and 啊，还好我考我眼睛瞎了，我电脑旁边了。啊，这是负的吧？哦，对不起，我靠，那很抱歉，我害怕它有那个镜子。
Groups are almost done. I'm going to be uploading some of your results soon. But before we move further, remember you place back all materials, the antibiotic, this with your vials, back into the pet conditions here. Make sure that you've returned everything that you've used. And if you use any extra tubes, you'll place them down at that black tree near the right side of the incubator. Also, for the catalase test, some of you may be wondering, you're seeing a lot of E. coli. You're seeing a lot of bubbling for Staphylococcus epidermidis. What does that signify? Do you think it's catalase positive or negative? Okay, positive. Three positive. Yep. Perfect. So now, when it comes to E. coli, you may have seen little bubbling or slow bubbling. That is essentially an indication of it having minimal propensity of catalase, and you could say in terms of background limitations or background information that it's essentially catalase negative. So just bear that in mind when you're recording your results in your post lab or with your discussion. And by the way, it's very important for actually detoxification, because if you have hydrogen peroxide in bacterium, for example, it actually can be quite toxic. So I want you to think about the applications of this. Exercise 3 looks into penicillin, right? Because if you think about the ampicillin or the penicillin family and the many antibiotics that could actually essentially cleave the beta-lactam ring, then you want to think, let's say as a medical practitioner, if you have an individual with a specific disease or bacterial infection, which of the appropriate antibiotics would you use? And as a bit of homework, I want you to start thinking about which antibiotics serve particular functions and why you would expect down the road, if you can predict, which antimicrobial site or slide would appropriately essentially give you unique results. Oh, this exercise 5 is just going to be done virtually on the slides here. Now, is your group done? Almost done exercise 1? Yep. Give me 1 moment.
It says to do that and stuff.
Your unknown is actually it's more like your E. coli and Staphylococcus epidermidis. So you could use the same, like on the slide, you could use the same bacterium that you had before from your solid agar. Just add in small, like swab in a given amount of your bacteria.
From the previous graph,
No, 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 not from your unknown assignment. It's of E. coli and Staphylococcus epidermidis.
Well, that's just like before.
Yeah, see these two. So these are like your it's not necessarily unknown, but potentially this is what your task is of Staphylococcus epidermidis versus E. coli.
We're just determining the
Yeah, yeah. Krista, you need that pipette for a particular reason. Reason being is because if I'm not mistaken, it's group threes and they need it for exercise one. If it's okay. Like there's one more good time. But they should you guys have pipette? We can. There's just one more group.
Okay.
We'll take care of it. If you guys are cleaning up, that's okay.
Okay.
No,
That's one. But I wanted to get it up. It's for the keeping.
Like each group should have one. See there, have grab the pipette from the side of the thing.
oh, yes. Like, I haven't looked at it.
Okay, so in about a minute, I'll talk about the hand washing exercise. I need group one to please come as soon as possible. So that way, as a group, we can go over all the material. By the way, folks, hopefully in due time, after the hand washing exercise, I want you to all upload your results for the viable counts. Make sure you take the means, the averages of your values for colonies that are 30 to 300 for each time point across your dilutions. For which one? No, well, we're going to talk about it because it's actually for your generation time assignment. Where did you get them from? Remind me? Oh, you can, let me see where it's best. Return it back. You hopefully still have that the paper, right?
Yeah.
So return it back. Just keep it away from the tray. So that way people don't think it's autoclaved already.
Okay.
But you no problem. Any final questions before I move further? Let's move to, let me just see if I missed anything. Beta-lactamase once again breaks down the beta-lactam ring in antibiotics. So hopefully you understand this from the penicillin family. Ampicillin as an example, penicillin as we've also touched upon today. And nitrocephin is actually essentially your assay of how it's going to be able to react and thereby deduce with a change of color if it's catalase, beta-lactamase positive or negative, i.e. if it has the enzyme or not. Catalase, once again, very important for redox, essentially the removal of hydrogen peroxide and the ability to generate water and oxygen gas through fermentation. Hand washing, so this is done virtually, i.e. you guys are going to listen, share your thoughts and ideas and make notes in your discussion for the post lab. So back in the day, if you would, let's say, go into a lab as you came in today, you really have a few options in mind. You could choose cold water to wash your hands, hot water. You could use with soap accordingly and rinse multiple times. There's this video also, if you haven't seen, it's quite interesting because it can show you how much cross-contamination and perhaps how many bacterium you could really have on your skin. So just bear that in mind. Really, the plates are shown here. It's a fun exercise, but you can think about it for yourself. Just like how you would have done in the past, from our previous exercises, hot water, cold water, with sanitizer, following a rinse, or just with sanitizer, control, which by the way, is like you putting your hands into agar. So just to give you guys some perspective, you can see different types of bacterium that grow. And this is, by the way, like a blood agar test plate, which essentially would show you deep contrast, right, with the red background or foreground, with bacterium either appearing white, green, yellow, etc. And so let's just have an idea to appreciate, I guess, a little bit of hygiene for us all. Before hand washing, you could see some bacterium in panel 1. After poor hand washing, you still see bacterium. By the way, as we did in labs 1 to 2, we saw and we used different bacterium. Our unknown assignment, we saw 6 different types of bacterium, hopefully across a myriad of them. So the idea is, like, hypothetically, if we had the same bacterium here, we should be able to actually identify each of them accordingly based on what principles they have. For example, L. lactis would be catalase positive, capsule positive with skim milk only, right? So think about that table, think about your flow charts from the past, and hopefully you should be able to distinguish them accordingly. Now, after poor hand washing, you still see some bacterium. After proper hand washing, you still see a significant drop, maybe of 1 or 2 colonies, or maybe a few, as I could see here, and maybe, if I'm not mistaken, down on this side. So you guys could still see some bacterium, which could give you some indications of a few things. Maybe there's still some areas of your hands that you probably haven't washed, right? During COVID, we were probably given some protocol as how we could wash our hands for a few number of seconds, and between our palms, from the edges of our fingers, etc. And so think about that, because those are often weak spots where bacteria may actually grow accordingly, right? So bear that in mind as we leave our lab today as well. Wash your soap accordingly. But noticeably, you'll notice that cold water is actually often a good way to kill bacteria, not actually warm. And there can be reasons as to why warm water can work with different bacteria. And hopefully, my goal is that in a week or 2, you're going to actually appreciate this far more better, because we're going to likely focus on different bacteria with different temperature conditions and how they best grow. So that's where you're going to appreciate, thermophiles versus homeophiles or halophiles and cryophiles, and essentially the different temperatures that bacterium grow well in. Maybe some of you remember 2-3 years ago going to AO2 and seeing different different organisms growing in different environments, right? So just like how organisms thermoregulate, bacterium do the same. And so think about this as it relates to your host response, a host bacterial relationship with the different bacterium that could grow. Quite anomaly, after a cough as well. So just think about, the bacterium that could grow, and not all of them are, harmful. Some of them grow in the gut flora, which probably Dr. Brun has pointed out to you guys. Hundreds of millions, if not billions, of bacterium that grow within our bodies, and they're beneficial for ourselves. And so think about, the importance of sustaining them, proliferating them at the right amounts, with the right generation time considered. And then hopefully, you guys will able to appreciate the effects of the bacterium in a myriad of ways. Exercise, group 1, are we almost done? Yeah, can we have it added into the microplate? Perfect. So now let me talk about generation time, the assignment. Any final questions, by the way, regarding exercise 5? Yeah? Okay. So for the generation time assignment, I've touched upon this on Quercus. I really hope everyone's seeing my messages on the inbox, because I try to give very detailed information where needed, because of wanting to make sure that you guys can succeed throughout the course. And we have 23 minutes, so hopefully we could go through this in due time. I just want to highlight, once group 1 is done, we'll add them into the microplate. But the idea is generation time is last week's material, and the Bible comes from this week. Meaning, from the microplate reader results that I uploaded on Quercus for Practical 2, which you should see labeled at 37 degrees, you should be able to, and let me show you the Word doc that I gave as a guide. Not pancakes. Just okay. Ooh. Okay. Let me go back here actually, just to go with the slides to make it easier. You guys know that on Quirkus you saw semi-log paper, right? Either from Dr. Brunt's module or from my own. Essentially, this is for you to prep for the generation time assignment because I really hope everyone's paying attention. Generation time really needs you to focus on how long it takes for your bacterium to double. And you see this best through semi-log paper because the increments on your y-axis, be it of turbidity, of absorbance values from your microplate reader, or from the number of cells, i.e. the cell density from your viable counts, they're done in the y-axis. So if you see a doubling, what you simply could do on semi-log paper, right, from what you've seen and what you could see here in the last slide, is let's say, I want to make sure if the okay marker is stable. Let's say this is your semi-log paper, also in green. You have to label everything, right? So please make sure I do keep this in mind that let's say your x-axis, as you know, is in temperature, sorry, time. So your time point technically starts at 0. I know some of you asked about, seeing it at 14 minutes or so. The idea is that based on the Excel sheet, you're taking your first reading of values, not of, I should show this to you guys in case you have problems with this. So let's say MS2. I'll actually show it here, microplate. We'll show it with practical 8. It's very similar. So you see these results. Hopefully everyone's paying attention. I want you to consider taking read 2 as time point number 1, right? So go down because you should have 9 time points in total, which is going to go from here, time point 1, with the values that you see here. And as I showed on the, oh, it's already open. Viable counts. Okay. Okay, I'm going to open both so you guys can see both. The idea is that, and I try to make this easier with a guide so that way you guys can organize it for yourself. You could print it, write it out, and then upload, or sorry, make your graphs accordingly for the FLR. Sorry, for your generation time assignment. The following is what you need to do. Hopefully everyone can see it on this side. Remember those values that I showed you. So you pretty much have all of this provided, right? For each group. Group 1, this is just a demonstration, so you actually, for all groups, have to update this accordingly. But the idea is, as an example, you have your blank, you have your reps, 1, 2, 3, and then your mean absorbance is what you're going to calculate, right? All of these values are essentially ones that you derive afterwards. So all of this, you're going to copy and paste from Excel for your practical, and you're going to repeat this for at 37 degrees and at 30 degrees. 30 degrees, and by the way, we're practical 2, right? But for practical 1, they're doing 30 degrees. So you're going to repeat this across all time points, across 37 degrees and 30 degrees for plotting. But I'm trying to make it simpler by you just essentially reading the instructions above. And hopefully this could suffice because you do have 2 weeks to do this, and it's not too difficult actually. The idea is that you have the values right near the bottom here. You have the raw values. I need you to essentially calculate the mean absorbance of the first 3 reps. Then correct, i.e., so like your average, you got the average now for 3 reps here. You're going to subtract your average, the mean absorbance value, by the blank. That's going to be correcting for the turbidity. And your result, you're going to do this for all groups per time point, right? And that's what you're going to treat as time 0. And you're going to do this as your lab, I call it your lab average, or your mean absorbance value corrected. for turbidity. And don't forget your units is OD 550. So you're going to be doing that for all time points. You repeat that for essentially, and it's pretty much you have this data already available. You're just copying, pasting it once again, but you're deriving these values thereafter, and you plot the final value, i.e. the one that's going to account for all corrected mean absorbances by their mean for the lab once averaged all together. You do this for each time point from 1 to 15, 30, all the way to 120, if I'm not mistaken, to 2 hours for microplate reading results. And you do that not just for our lab, but for the other lab, 30 degrees. I've uploaded all this material during reading week. So you guys should have enough time to do this. And I know some of you may be, wondering, okay, I have a plaque assay as well for FLR. What do I do about that? Before I touch upon it, I just want to highlight as well the viable counts. So let me just highlight this once again. Let's say these are your time points. You're going to put the label minutes, 0, 15, 30. They all should be even. So just count the number of increments across and then divide accordingly for the 9 different time points. 60 all the way down to 120, which is 2 hours. Now you're going to be adding in that final column, remember, once again, folks. So just bear that in mind with the mean. Essentially, remember what I said, the mean lab absorbance value, which you see there, and then in OD 550. On the slide, just remember to have this in brackets or 550 nanometers. Now, on semi-log paper, you go increments that are uneven. In fact, they're in a multiplicity. So if you have, for example, 0.01 here, it's going to go up to 0.1. Remember that. It's not going to go to 0.02. Please make sure group 1 especially, and all groups that you're all paying attention. Very happy we have this done. I'll come to this soon. I just want you to make sure that you pay attention to this because you'll see it also on the slide. But the exact same thing, see, like you see here, it starts at 1. I need you to all really keep an eye on this because some people make this mistake. If this is 0.01, this is not 0.02, this is 0.1. It's multiplying by 10 each time. So just bear that in mind. For absorbance readings, they're going to be more thin because as we just talked about, essentially they go 0.01, 0.1. The last one, i.e. technically what we see as 3 here, that's going to be 1 as an absorbance reading. Meaning that generally speaking, your range of absorbances from the microplate reader are going to be around this range, around here. Meaning, once you plot all the values with your mean absorbance value, you don't have to put error bars because you're doing this manually. But essentially, you should see something like this. Something like this. And then, for this task, and I know it may sound daunting, but you guys have done a lot very successfully. So I can imagine you guys do this also very successfully as well. The idea is that you are going to make a curve or a line, right? Smooth curves are nice, but the idea is that within the portion that you know that there's a slope, right? Like a slope that goes up, not down or just remains stagnant, that the location in which you know that the readings appear to have a linear slope, like the one I just showed, you have to do the following. And in the past, I've actually told people to do it 2 ways. You guys can do it both ways. I love it as well. In that you have a slope, you calculate the slope, meaning that you're going to go down to, your Y1, and that's your Y2. It could be any of those points, and then any of them in between. Just don't have them within 15 minutes for an incubation unless that's the only time that you have, because it usually isn't sufficient. You see how this goes over 15 minutes, maybe 20 minutes as an example. The distance between X2 to X1 is pretty much your generation time. Let me just clarify what I mean by that. If the difference between Y1 and Y2 is exactly double, then this is where option 1 for calculating generation time comes in to determine the distance between x2 and x1. What you normally do, as shown there on the slide, is that you take any of the points on that linear slope, but you make note of the following. You're calculating the slope, right? And that revolves around you taking the log, because after all, we're on semi-log paper. So that means, just like how you do y equals mx plus b, where the slope, where n is like y2 minus y1 divided by x2 minus x1. You, instead of just doing this, you're adding in the log. I want to make sure everyone knows how to do log, because if you use a scientific calculator, quite simple. You just go on, for example, you press underneath D, log, you press any value, let's say in our case, 0.5, and you get a value. Normally, your values should essentially become positive as a slope, right? Like, it's understandable if this is negative and this is also negative, but at the very end, this entire equation should be positive, i.e. log y2 should be greater than log y1. So just bear that in mind. There's two steps once again. You calculate the slope as shown by the formula here, making note of log in your calculations for y2 and y1 divided by x2 minus x1. Once you've done that, that's your slope. But for determining generation time, you need to divide 0.3 by the slope. Does that make sense for everyone, by the way? Because that's very important. That's pretty much the main component for determining the slope. The other option, as I said, the second option is if technically y1, let's say 0.12, and then y2, for example, is 0.24, then if you know the distance between the two on the x-axis, that's your generation time. And what I want you to do is, because after all, this is your results, having everything labeled, the units, the legend, by the way, not just for your, like, for our practical, but for the second practical as well. The same is very similar for your viable counts, which I'll get from you guys soon, or will be, yeah, from you guys soon. But importantly, you have your intro. I've talked about this in the inbox. Just have a look. You're talking about generation time. Very generally, it's about 3 sentences, 3 to 5 sentences with your research question or objective of the lab. Then you go to your results. Don't have methods. Just focus on the results. And then do what I've shown here for the 4 curves that I've talked about. Remember, 30 and 37 degrees, viable counts, and microplate reader absorbance results. That's all 4 curves that you would do for this, making sure that you could do them technically on the same, like as an idea, because I know it may be too much to bear. The idea is that you could do, let's say, viable counts on this side, right, on this edge, because it goes from 1 to 10 here. And then this technically goes from 1 to 10 there for the next graph. So to make it easier, microplate reading could even be done here. Viable counts can be done here. Either way, just make sure that you fit everything accordingly. The viable counts, as well as microplate reading results, you need to have units. You need to have a figure legend, because you're dealing with 4 different curves across temperatures and techniques. And you need to have a figure caption, a figure caption that's detailed. Try to have it specific. It could be a title, that's okay. But I need it to be specific with details of the species, the bacterium, the technique, the conditions. So, and the units if you can as well. That would be greatly appreciated. And then on the reverse side of your log paper, you could show the calculations as to how you determine the slopes, which are 4 different techniques. That's what you're submitting with me with your intro. And then at the end, inshallah, you're going to show me your discussion, discuss a few things that are very important, just like what I touched upon for the FLR, which you're going to see briefly for your plaque assay. You talk about your objective once again. You talk about what your results are. You discuss if your results are expected or not, and describe why if not, with your limitations. And then thereafter, the extensions. For the generation time assignment, by the way, because it is Okay, I'll give you some advice regarding for the generation time assignment. Bear in mind that there is a generation time of E. coli. So search it up online, see what you can find for 37 degrees or 30 degrees. See if you can compare the values and why, if it's not similar, why it is or why it isn't. But I will say, bear in mind that you're placing close attention to your calculations, and in the discussion, also include the implications. That pretty much highlights the generation time assignment. Are there any final questions regarding this? Pretty much what I just said to you now is right here. So think about what I've touched upon. Any questions regarding this? It's slide 2 of week 6. So if you have any questions, come back here again as well. Yes. It is in the files, but I do want to make sure you guys are paying attention because a lot of these questions may come up again, and it takes up a lot of time for myself as well for the different practicals. So just try to take note if there's anything that you've missed accordingly. Perfect. I think there was a question. Yes. Yes. So you need to print it out. I would recommend printing it out maybe after today. At the library. If you need financial assistance, by the way, just let me know. I'll print it out for you guys. Yes.
So just to be clear, we're manually doing the semi-log graphs on the semi-log paper, but we're writing bigger captions, the introductory, and the including stuff in our lab notebook.
No. So great question. Everything related to the results, including calculations for generation time, are done on the semi-log paper. The calculations themselves can be on the other side because it's normally blank, but the semi-log paper should include essentially this material, construct 2 semi-log graphs, to essentially include the plate meter and the Bible counts for both 30 and 37 degrees for both sides. So essentially consider everything that I said here, including your units and the figure caption or title on your semi-log paper. The intro, your calculations will be on the other side of the semi-log paper, but your intro and conclusions can also be done on the other side to make it conservative, not to waste paper. So like the idea is like have everything labeled, your graph, what you need for raw, for results. All of that material is on these 2 slides. Sorry, on these. Yes, on the graph itself. The calculations can be done on the other side as further results for your calculations. Above it could be your intro. Beneath it, your discussion / conclusions. That's pretty much it. Before I end off, hopefully all of you can either send to me your viable counts today. I can record them afterwards if you're in a rush. The other thing is, so the Plaque Assay. I want to ask how many of you have problems with it, or actually before I do this, because we are on a bit of time, and I want you to get it done as soon as possible. Okay, so just before we leave, the Plaque Assay, you know your results. Before though, any other questions about post-lab, pre-lab for this week or of the generation time assignment? Any questions? Yeah. So pretty much okay. Whoever wrote it down, perfectly. Pretty much having it on the top. Normally you have captions that are very detailed. However, because you have limited space, I don't mind you having it on the top as a title, but the title has to be descriptive. For example, it needs to include the technique, the species, the temperatures that you're dealing with, and any other additional material like the units of your measurements. Now, on the other side, you can include your intro, the calculations within your results, and the discussion. And then you would submit them to me whenever you complete. But the final day of submission is on week 8. So at the very beginning of the lab, I actually have to take it from you. Please make sure that you actually bring it to me, because otherwise you don't want to actually be penalized for late submission. So just bear that in mind. 2 weeks from now.
That was next week. No, sorry. What month is it? March.
I see. Yes. So that is week 8, actually.
Oh, I lied.
No problem. Any other questions? Let me just briefly go over the plaque assay. I wrote it down here for those that want to know. Briefly, once again, I just want to highlight it's similar to the generation time assignment, but there's more detail that is required because this is a formal lab report. You need to have obviously a descriptive title, an abstract. The materials are already in the outline, so just don't worry too much of what I'm saying now, but make sure you have all materials that are required. I need, though, something important. Your plates need to have figure captions, as I've talked about for the pre-lab today. Make sure for your results, even for today, for your post-lab, you have descriptive figures. You have them either as photos. Actually, you should have them as photos for the FLR. If you don't have them already, see the ones that you have on Quirkus. Make note of actually writing the calculations of determining the plaque assay results, meaning that once again, if you have, and this is just transformation material, if you didn't include it for the post-lab for week 5, make a note of these materials as well. But the idea is the following. For the plaque assay, you have 5 different plates, A to E. Now, for the colonies between 30 and 300, you need to actually determine that you have to count them accordingly, right? So if you have 242 and your dilution factor for all of them is, let's say, for the one I'm looking at right now, 10 to the minus 5. You're going to divide it by 10 to the minus 5, meaning that you're multiplying by 10 to the positive 5. And that 242 times 10 to the power of 5 is going to be 2.42 times 10 to the power of 7 PFUs. So you're counting the holes in your plates. Count them up, multiply by the inverse dilution factor, the final one of your plates, which is like the positive exponent. And essentially, do this for all of your plates. Once you do this, because you have four plates that are experimental, non-control, and so essentially you need to take the average of all 4. If you have 3 plates that are within that range, 30 to 300, that do the mean for those 3. But importantly, you may have some that are too many, over 300, some that are too few, less than 30. So you can omit them from the calculation, but describe them in your result. I need to know that. So that way I know that you were attempting to count at least 4 different plates. And then with the 30 to 300 colonies, take those as your mean for determining the number of PFUs. I hope that helps, because that's what you really need to describe per group, individually within your group's plates. And then with your discussion, briefly describe the objective, results, describe the limitations and extensions. And I have some points here. You can have a photo of any other material. I do want to end the lab, but if there's any final questions, let me know. If anyone wants to stick by to give me the viable count material, also let me know. With this, really appreciate your help and the conclusion of today's lab. Make sure, once again, you return back all materials. The plates, if unused, return back. The discs need to be returned to the front in this tray. Here's that in the rifle. Definitely needs to be returned as well. Please bring this back. There is a missing BD box. I need this to be returned. Make sure for precaution 4 to 7 days, physiological test, you can throw them into the garbage. Honestly, and any material that is touched bacteria, throw them into the autoclave, not the glass package itself, just the disc, small disc for antibiotics, the slides, semi-chrome, into the autoclave as well. The beakers, then essentially any liquid waste goes back into the Westgadine with the pipettes as well. I can have
Yeah.
So as a tip, you guys could look into the life cycle as an example because that relates quite readily to the bacteriophage and Plaque Assay assignment or SLR. So you can talk about that, go down to bacterium and to the bacteriophage, and then into your research. If it has to be specified in the outline, more. Yeah. Something is in need of all four, because essentially we have let's say this because UNCF is not going to be a right? Once we get a value, multiply the column by the you're going to take that TFP reading for each plate and get the mean of all four. That's but I need you to reportedly know that the observation for all places. Make sure that you include observations for all 5 places, including the control. Calculations are at max up to 4. They could be 2 to 4 plates. If you find that there are so many to count EMTC, to count EFTC as your observations for specific plates. Apart from that, make sure to wash your hands. That's exercise 5. Have a great day. Take care, everyone. If you guys can record and send to your PFUs, that would be also really appreciated. Each group, please make sure for your PFUs, Plaque Assay, please make sure that you send me your data so that way I can actually incorporate it for the entire practical. That'll make things easier actually.
Yeah. Oh, I don't think that's time to play.
If anyone maybe takes this there and target,
You're trying to get it.
if
I'm gonna try to
no, it's okay. It's okay. I don't think it's okay because I actually started this account. Okay. I appreciate your time. But if anyone from the lab, these clips belong to them, take them now because in case you actually want to take photos, And we should do them before they're exposed. I don't want, can you see what name of the letters? Okay. I just don't want to Grade 12, please send me your plot data, your Bible account, and I could upload them again. Thank you. Take care. Have a great week. That's great. Okay.
Thank you. Bye.
But the idea actually, it's the idea that we talk about. I know here, outside of the generation, we have to include specific music that we will take the top of the ball. So let's say it's the original solvents back here. Yeah, it's so fun. And drawing I would recommend those with the deeper action than the results. That should suffice. I practically did also small one, like what are conclusions based on the action. If you have based on different use action, you can do that. That would be a reason. Do that, I mean, not, but it's also good to have a lot of that we know that they are factors. Especially representative figure. It's like that's let's say if you're the sole figure of your alarm, I can just like looking at that to get a comfortable idea as to what we did, how we did it, and what the results are, and what that means. So it's like you do that so like let's say like high that's like that's the point of the results there. Like the reason I think especially is some other areas on the road or other practices require more road process. So that's why like it makes sense that like maybe ask you to start with limitations to that I try to like I just want you guys to be as discreet as possible, like reduce, and that way, like you get the best representation.
So like, should we say like figure A is control, figure B is the
Yes, yes. I agree. I would actually recommend doing that. I'm just gonna shut off.
You also want us to mention the counts in the figure or just
If you can, you can put the final cell density, pack density. Like for each, you can actually put the value. You don't have to put like you can just put the value for each plate. BTE. Now also they have control of that density price, or what the control is. But B, D, C, D, E, you want to put the value of the final calculation. That should be everything done by the everything.
what did they use to classify for?
Some of them use some of the plastic pipettes. Some of them may have used it for the microplate. Because it's much easier with the I think microplate. Yeah. Putting up rooms that I've highlighted here, also be helpful. Some new results of population there to provide values and population value, and then to put into your limitations. That includes like cross contamination, that can include like limited volume that needs to be done. But or you can fire it. But I hope and pretty much most questions I hope you guys got to enjoy the lab because we did finish well. But I just want to make sure you guys are aware. I know sometimes for also it's just like just I know Plaque Assay, results, the lab of chemical and so I'm trying to like be observant of that. So that way you guys don't have to feel rushed. Yeah. You guys can go. Thanks a lot for your time. If you have questions, please. Hopefully, I think it's okay. I don't like because I'll be honest with you, a lot of things are a bit intense. So that's why it may take a bit of time for me to respond. But overall, I do try to make sure that I can essentially be fair and marking you guys. Some like I don't want to be comparative, but I generally speaking try to be both generous and just. So that way you guys can like feel rested and reassured that I'm not like being biased or penalizing you in ways that wouldn't be just essentially. But I also like to count for different things. So hopefully do your best. Assalamu alaikum wa rahmatullahi wa barakatuh. Alhamdulillah Rabbil Alameen wa Rabbil Arshil Kareem. La ilaha illa ant. Subhanaka inni kuntu minadh dhalimin. Ya Rabb. Alhamd. Subhanallah Rabbil Arshil Kareem. We are actually about into our 19th seminar here in our Golden Lab, here by BIOC17, week 6, group 1, inshallah ta'ala. So I just want to say, for those that are attending, listening online, inshallah ta'ala, students are, going through a myriad of different exercises, as you can hear. I haven't specified them to you all directly, but essentially there were 5 exercises for today, including a virtual hand washing one. But they went through different things. They went through antimicrobial susceptibility tests. They had 2 other additional tests, cephinase and beta, and a catalase test. And the first one was also very beautiful. I'm cleaning up at the same time. But inshallah ta'ala, I do hope that students and like yourselves are actually being able to appreciate holistically the different effects. For generation time, which is upcoming assignment 3, but also the plaque assay due up soon. And so inshallah ta'ala, students will also be able to exercise and learn how they could best apply the material that they've gained from the lab and into the implications, limitations, and extensions of the lab to the world outside, right, outside of the lab environment. So inshallah ta'ala, with this, may Allah accept all of your efforts. Brothers and sisters, stay up to date. Inshallah ta'ala, we have many things that are in store. We also have our Golden Quran series for Ramadan. And so inshallah ta'ala, you'll see just for, or many just hopefully with selected ayat reflected. And maybe we'll just say, to make it simplified, selected ayats across the Noble Quran Kareem Aziz in due time, inshallah, during this blessed month of Ramadan. Ramadan Mubarak to all that are listening. May Allah subhanahu wa ta'ala make it easy for you, your family. May Allah subhanahu wa ta'ala accept it from us and allow us to reach and maximize our best potential yet. The Jannatul Firdaus al-A'la, from Laylatul Qadr to the next, and the many more Ramadans, and for the rest of our lives, really. Ameen ya Rabbil Alameen. Barakallahu feekum. Wa alaykum wa rahmatullahi wa barakatuh. Assalamu alaikum wa rahmatullahi wa barakatuh.